Why We Die: The New Science of Aging and the Quest for Immortality by Venki Ramakrishnan explores the fundamental biological processes behind aging and death and examines current scientific efforts to extend human lifespan. Ramakrishnan, a Nobel Prize-winning molecular biologist, provides a comprehensive overview of aging research, from evolutionary theories to cutting-edge experiments. The book begins by addressing our innate fear of death and the various ways humans have tried to cope with mortality throughout history. Our awareness of death is unique among animals, requiring a level of self-awareness and cognitive ability that emerged relatively recently in evolutionary terms. Death is a complex biological process that occurs at multiple scales, from individual cells to entire organisms. At its core, aging results from the accumulation of damage to our molecules, cells, and tissues over time. This damage eventually leads to the breakdown of bodily functions and, ultimately, death. One of the key concepts in understanding aging is the evolutionary theory behind it. Natural selection favors traits that enhance reproductive success early in life, even if they may have negative consequences later on. This is because, in nature, most organisms don't live long enough to experience the detrimental effects of aging. Several important evolutionary theories of aging help explain why aging occurs. The mutation accumulation theory proposes that harmful genetic mutations that only manifest in old age can accumulate in populations because natural selection doesn't act strongly against them. For example, a mutation that causes a fatal disease at age 80 wouldn't be selected against in a population where most individuals die before 40. The antagonistic pleiotropy theory suggests that some genes beneficial in youth may become harmful later in life. A classic example is the p53 gene, which helps prevent cancer in young individuals, but can contribute to aging by promoting cellular senescence. The disposable soma hypothesis posits that organisms must balance energy investment between reproduction and bodily maintenance. This theory explains why animals that reach sexual maturity quickly and produce many offspring tend to have shorter lifespans than those that mature slowly and produce fewer offspring. These theories help explain why aging occurs, but they don't fully account for the wide variation in lifespans among different species. Some animals seem to defy the usual rules of aging, living much longer than we would expect based on their size or metabolic rate. For instance, naked mole rats can live up to 30 years, which is extremely long for a rodent their size. They show remarkable resistance to cancer and maintain healthy protein function well into old age. Studying the molecular mechanisms behind these traits could provide insights for human longevity. Bowhead whales can live over 200 years, making them one of the longest-lived mammals. Their longevity may be related to their ability to repair DNA damage and resist cellular stress. Some species of clams and tortoises can live for centuries, with some individuals estimated to be over 500 years old. These long-lived species are like nature's experiments in extreme longevity. Just as engineers might study a car that lasts for 1,000,000 miles to improve the design of other vehicles, biologists hope to uncover longevity tricks from these animals that could be applied to human health. The book delves into the molecular mechanisms of aging, starting with DNA damage and repair. Our genetic material constantly faces threats from both external sources, like radiation, and internal processes, such as errors in DNA replication. The cell has evolved sophisticated mechanisms to detect and repair this damage, but these systems become less effective with age. DNA damage and repair play a crucial role in the aging process. As we age, our cells accumulate more and more DNA damage, which can lead to mutations, cellular dysfunction, and eventually, age-related diseases. The cell has multiple DNA repair mechanisms to deal with different types of damage. For example, nucleotide excision repair removes bulky DNA lesions caused by UV light or certain chemicals. Base excision repair fixes small chemical alterations to DNA bases. Mismatch repair corrects errors made during DNA replication. Studies have shown that mice engineered to have defective DNA repair mechanisms exhibit accelerated aging. For instance, mice with mutations in the XPD gene, which is involved in nucleotide excision repair, show premature aging symptoms including osteoporosis, neurodegeneration, and reduced lifespan. Conversely, enhancing DNA repair capacity can extend lifespan in some model organisms. For example, increasing the expression of SIRT6, a protein involved in DNA repair, can extend lifespan in mice by about 15%. DNA can be thought of as the blueprint for cellular function. Just as a building constructed from a damaged blueprint would have problems, cells operating with damaged DNA struggle to function properly. Over time, accumulated DNA damage can lead to cellular senescence, cancer, or cell death. Maintaining genomic stability is a key focus of anti-aging research. Strategies being explored include enhancing natural DNA repair mechanisms, developing drugs that can protect DNA from damage, and even using gene editing technologies to correct accumulated mutations. Another crucial aspect of aging discussed in the book is the role of telomeres. Telomeres are protective structures at the ends of chromosomes that play a crucial role in cellular aging. These repetitive DNA sequences act like the plastic tips on shoelaces, preventing chromosomes from fraying or sticking together. Each time a cell divides, its telomeres shorten slightly. When telomeres become too short, the cell stops dividing and enters a state of senescence or undergoes programmed cell death. Researchers have found that cells from older individuals generally have shorter telomeres than those from younger people. In some premature aging syndromes, telomeres shorten more rapidly than normal, leading to accelerated aging. An enzyme called telomerase can extend telomeres, and it's active in certain cells like stem cells and cancer cells. Some researchers have explored activating telomerase as a potential anti-aging strategy. However, this approach carries risks as unbridled telomerase activity could promote cancer. Telomeres can be thought of as a burning fuse on a candle. As the fuse burns down, telomeres shorten. It gets closer to the end of its life. Just as you can't easily relight a burnt-out fuse, cells typically can't regrow their telomeres once they're critically short. While the discovery of telomeres and their role in aging has been a major breakthrough, Ramakrishnan emphasizes that telomere shortening is just one of many factors contributing to aging. Simply maintaining long telomeres is not a guaranteed path to longevity. The book also explores epigenetic changes that occur with age. Epigenetics refers to modifications to DNA that don't alter the genetic sequence but affect how genes are expressed. These changes accumulate over time and can significantly impact cellular function. Epigenetic changes include DNA methylation, the addition of methyl groups to DNA, and histone modifications, changes to the proteins that DNA wraps around. These modifications can turn genes on or off, affecting how cells function. Interestingly, some of these epigenetic marks can be used as a biological clock to estimate an organism's age more accurately than chronological age alone. For example, Steve Horvath developed an epigenetic clock based on DNA methylation patterns that can accurately predict biological age across multiple tissues and species. Studies have shown that manipulating epigenetic marks can influence lifespan in model organisms. For instance, increasing levels of certain histone modifications can extend lifespan in worms and flies. Epigenetics can be thought of as the software running on our genetic hardware. Just as software can be updated or become buggy over time, our epigenetic patterns change throughout our lives, sometimes in ways that promote aging. The reversible nature of epigenetic changes makes them an attractive target for anti-aging interventions. Some researchers are exploring ways to reset epigenetic patterns to a more youthful state, potentially reversing some aspects of aging. A significant portion of the book focuses on cellular processes that contribute to aging. One key player is the protein quality control system. As we age, our cells become less efficient at identifying and removing damaged or misfolded proteins. This can lead to the accumulation of toxic protein aggregates, which are associated with neurodegenerative diseases like Alzheimer's and Parkinson's. Proteostasis, or protein homeostasis, involves the processes of protein production, folding, trafficking, and degradation. As we age, our cells become less efficient at maintaining proteostasis, leading to the accumulation of misfolded or damaged proteins. For example, in Alzheimer's disease, a protein called beta amyloid forms toxic aggregates in the brain. These aggregates are thought to contribute to the death of neurons and cognitive decline. Similar protein aggregation occurs in other neurodegenerative diseases, such as the accumulation of alpha-synuclein in Parkinson's disease. Studies have shown that enhancing protein quality control mechanisms, such as the heat shock response or autophagy, can extend lifespan in various model organisms. For instance, increasing the expression of heat shock proteins, which help other proteins fold correctly, can extend lifespan in worms and flies. Proteostasis can be thought of as the quality control system in a factory. As this system becomes less efficient with age, more defective products, damaged proteins, slip through, causing problems throughout the cell. Maintaining proteostasis is a key focus of many anti-aging interventions. Strategies being explored include enhancing cellular cleanup processes like autophagy, boosting chaperone proteins that help other proteins fold correctly, and directly targeting misfolded proteins associated with specific diseases. The book explains the importance of autophagy, a cellular recycling process that breaks down damaged components and recycles their building blocks. Autophagy declines with age, contributing to the accumulation of cellular damage. Enhancing autophagy has shown promise in extending lifespan in various model organisms. Autophagy is a process by which cells break down and recycle their own components, including damaged proteins and organelles. It's crucial for maintaining cellular health and has been implicated in many aspects of aging. Studies in model organisms have shown that enhancing autophagy can extend lifespan. For example, increasing the expression of autophagy genes in worms can extend their lifespan by up to 50%. In mice, drugs that enhance autophagy, such as rapamycin, can extend lifespan and delay the onset of age-related diseases. Autophagy declines with age, which may contribute to the accumulation of cellular damage. This decline has been linked to various age-related diseases, including neurodegenerative disorders and cancer. Autophagy can be thought of as a cellular recycling and cleaning service. Just as a city with an efficient waste management system stays cleaner and healthier, cells with robust autophagy can better maintain their health and function. Enhancing autophagy is a promising strategy for anti-aging interventions. This can be achieved through various means, including certain drugs, dietary interventions like caloric restriction, and exercise. The book also explores the role of mitochondria in aging. These cellular powerhouses produce energy, but also generate harmful byproducts called reactive oxygen species. The accumulation of damage to mitochondrial DNA over time can lead to dysfunction and contribute to the aging process. Mitochondria are crucial for cellular energy production, but they also play important roles in cellular signaling, apoptosis, programmed cell death, and the regulation of metabolism. As we age, mitochondrial function declines, contributing to many aspects of the aging process. This decline is due to a combination of accumulated damage to mitochondrial DNA, decreased ability to remove damaged mitochondria, and reduced production of new mitochondria. For example, studies have shown that mice engineered to accumulate mitochondrial DNA mutations at a faster rate exhibit accelerated aging. Mitochondrial dysfunction has been linked to various age-related diseases, including neurodegenerative disorders, diabetes, and cardiovascular disease. For instance, declining mitochondrial function in neurons is thought to contribute to the cognitive decline seen in Alzheimer's disease. Mitochondria can be thought of as the engines of our cells. Just as an old car engine becomes less efficient and produces more pollution, aging mitochondria become less effective at producing energy and generate more harmful byproducts. Maintaining mitochondrial health is a key focus of anti-aging research. Strategies being explored include enhancing mitochondrial DNA repair, improving the removal of damaged mitochondria, mitophagy, and stimulating the production of new mitochondria. For example, certain forms of exercise have been shown to stimulate the production of new mitochondria, potentially contributing to the anti-aging effects of physical activity. Ramakrishnan discusses several promising areas of research aimed at slowing or reversing aging. One approach focuses on manipulating nutrient-sensing pathways, such as the insulin IGF-1 pathway and the TOR target of rapamycin pathway. Reducing the activity of these pathways has been shown to extend lifespan in various organisms, from yeast to mice. The insulin IGF-1 insulin-like growth factor pathway and the TOR pathway are key nutrient-sensing pathways that play crucial roles in determining lifespan. These pathways evolved to help organisms respond to changes in nutrient availability, but in our modern environment of constant food abundance, their chronic activation may contribute to aging. Reducing the activity of these pathways has been shown to extend lifespan in various organisms. For instance, mutations that reduce insulin IGF-1 signaling can dramatically extend lifespan in worms, flies, and mice. In humans, certain variants of genes involved in this pathway are more common in centenarians, suggesting they may contribute to longevity. The TOR pathway regulates cell growth and metabolism. Inhibiting this pathway, either through caloric restriction or drugs like rapamycin, has been shown to extend lifespan in various organisms. For example, studies in mice have shown that intermittent rapamycin treatment can extend lifespan by 10 to 20% and improve various measures of health in old age. These nutrient-sensing pathways are like cellular fuel gauges. When they detect plenty of fuel, nutrients, they signal the cell to grow and divide. When fuel is scarce, they trigger conservation modes that can promote longevity. By manipulating these pathways, researchers hope to trick cells into a longevity-promoting state without actual nutrient deprivation. While these findings are exciting, translating them into safe and effective human therapies remains challenging. The insulin IGF-1 and TOR pathways are involved in many crucial biological processes, so altering them could have wide-ranging effects beyond just lifespan. Caloric restriction, or reducing calorie intake without malnutrition, has been known to extend lifespan in many species. The book explains the molecular mechanisms behind this effect and discusses the potential for developing drugs that mimic the benefits of caloric restriction without the need for dietary changes. Caloric restriction involves reducing calorie intake by about 20 to 40% while maintaining adequate nutrition. This intervention has been shown to extend lifespan and health span in various organisms, from yeast to primates. Studies in mice have shown that caloric restriction can extend lifespan by up to 40% and delay the onset of age-related diseases. In humans, short-term studies have shown improvements in various biomarkers of aging, including improved insulin sensitivity and reduced inflammation. Caloric restriction works by triggering various cellular stress response pathways that enhance repair and maintenance processes. It affects multiple systems in the body, including reducing inflammation, improving metabolic health, and enhancing cellular cleanup processes like autophagy. We can think of caloric restriction as putting the body into a resource conservation mode. Like a city reducing energy consumption during a shortage, the body activates various protective mechanisms when calories are scarce. While the benefits of caloric restriction are well established in animal models, its long-term effects and practicality in humans remain subjects of debate. Many researchers are working on developing caloric restriction mimetics, drugs that could potentially provide the benefits of caloric restriction without the need for dietary changes. One such drug that has garnered significant attention is rapamycin. Originally discovered as an antifungal agent, rapamycin inhibits the TOR pathway and has shown promising life-extending effects in multiple species. However, Ramakrishnan cautions that long-term use of rapamycin in humans may have significant side effects, and more research is needed before it can be considered a safe anti-aging intervention. Rapamycin was originally discovered in a soil sample from Easter Island and was found to have immunosuppressive properties. Later, it was discovered to inhibit the TOR pathway, which plays a crucial role in regulating cell growth and metabolism. Studies in mice have shown that rapamycin treatment can extend lifespan by 10 to 20% and improve various measures of health in old age. Remarkably, these benefits were seen even when treatment was started in middle-aged mice, suggesting that it might not be too late to intervene in the aging process. Rapamycin works by inhibiting the TOR pathway, which shifts cells from a growth-promoting state to a maintenance and repair state. This mimics some of the effects of caloric restriction at a molecular level. We can think of the TOR pathway as a master switch that tells cells whether to focus on growth or maintenance. Rapamycin essentially flips this switch to maintenance mode, potentially slowing the aging process. While rapamycin shows promise as an anti-aging intervention, its use is not without challenges. As an immunosuppressant, long-term use could increase susceptibility to infections. Additionally, as the TOR pathway regulates many crucial processes, inhibiting it could have unintended consequences. More research is needed to determine the optimal dosing regimens and long-term effects in humans. The book also explores the potential of stem cell therapies and regenerative medicine in combating aging. As we age, our stem cell populations decline in both number and function. Researchers are investigating ways to rejuvenate stem cells or use them to repair age-related damage in various tissues. Stem cells are undifferentiated cells that can develop into various specialized cell types. They play a crucial role in tissue maintenance and repair throughout our lives. However, as we age, our stem cell populations decline in both number and function, reducing our body's ability to repair and regenerate tissues. This decline in stem cell function contributes to many aspects of aging, including reduced tissue regeneration, impaired wound healing, and decreased immune function. For example, the decline in hematopoietic stem cells, which give rise to all blood cell types in bone marrow, contributes to the weakened immune system often seen in older adults. Studies in mice have shown that transplanting young stem cells into old animals can improve various measures of health and in some cases extend lifespan. For instance, transplanting young hematopoietic stem cells into old mice can rejuvenate their immune system and extend their lifespan. In humans, early clinical trials are exploring the use of stem cell therapies for age-related conditions like frailty. One study found that infusing mesenchymal stem cells into frail elderly patients improved their physical performance and reduced inflammatory markers. We can think of stem cells as the body's repair crew. As we age, this crew becomes smaller and less effective. Stem cell therapies aim to bring in reinforcements or retrain the existing crew to work more efficiently. While stem cell therapies hold promise, they also face significant challenges. These include ensuring the safety and long-term stability of transplanted cells, as well as ethical concerns surrounding certain types of stem cells. Researchers are exploring various approaches to overcome these challenges, including using a patient's own stem cells, autologous transplantation, and developing methods to stimulate endogenous stem cell populations. The book delves into the exciting field of cellular reprogramming and its potential applications in aging research. Cellular reprogramming involves reverting mature cells to a stem cell-like state, essentially turning back their biological clock. This technique, pioneered by Shinya Yamanaka, who won a Nobel Prize for this work, has opened up new possibilities for regenerative medicine and anti-aging research. Yamanaka discovered that by introducing just 4 specific genes, now known as Yamanaka factors, mature cells could be reprogrammed into induced pluripotent stem cells, iPSCs, with properties similar to embryonic stem cells. Partial reprogramming, activating reprogramming factors for short periods, has shown promising results in animal studies. For instance, studies in mice have shown that partial reprogramming can rejuvenate various tissues and extend lifespan without causing tumor formation, a risk of full reprogramming. One study found that short-term cyclic expression of Yamanaka factors in mice could extend their lifespan by up to 30% and improve various markers of tissue health. The treatment was effective even when started in middle-aged mice, suggesting it might not be too late to intervene in the aging process. We can think of cellular reprogramming like restoring a computer to factory settings. It erases the accumulated software glitches, cellular damage, and epigenetic changes, and returns the cell to a pristine state. While reprogramming shows potential for rejuvenating aged cells and tissues, controlling the process precisely to avoid unintended consequences remains a significant challenge. Researchers are working on developing safer and more controlled methods of partial reprogramming that could potentially be translated into human therapies. The book also addresses the controversial topic of radical life extension and its potential consequences. Some researchers and futurists believe that dramatic increases in human lifespan, perhaps even achieving immortality, may be possible in the future. These ideas range from more grounded approaches like combining multiple anti-aging therapies to more speculative concepts like mind uploading or cryonic preservation. For instance, gerontologist Aubrey de Grey has proposed a strategy called SENS, Strategies for Engineered Negligible Senescence, that aims to address 7 major categories of age-related damage. He argues that by periodically repairing this damage, we could potentially extend human lifespan indefinitely. Some tech billionaires have invested heavily in anti-aging research, hoping to dramatically extend human lifespan. For example, Google co-founder Larry Page helped launch Calico, a company focused on combating aging and associated diseases. However, most mainstream researchers, including Ramakrishnan, maintain a skeptical stance towards the more extreme claims of life extension advocates. They emphasize the immense complexity of biological aging and the many challenges that would need to be overcome to achieve radical life extension. We can think of aging as a leaky boat. Current medical approaches try to bail out water faster, treating age-related diseases. Radical life extension proposes to fix the leaks entirely, potentially allowing the boat to stay afloat indefinitely. The author also raises important ethical and societal questions about radical life extension. Would greatly extended lifespans exacerbate inequality? How would it affect population growth and resource consumption? Would it fundamentally change human society and our relationship with death? One of the most important distinctions Ramakrishnan makes is between extending lifespan and extending health span. Health span refers to the period of life spent in good health, free from major age-related diseases and disabilities. While increasing overall lifespan is a goal of much aging research, many scientists argue that extending health span is even more important. The aim is not just to live longer, but to remain healthy and active for a greater portion of our lives. For example, treatments that could delay the onset of age-related diseases like Alzheimer's, heart disease, and cancer by even a few years could significantly improve quality of life in old age, even if they don't increase maximum lifespan. A study published in the New England Journal of Medicine found that caloric restriction in non-obese adults improved multiple markers of health, including cholesterol levels, blood pressure, and insulin sensitivity. While the study didn't look at lifespan, it suggested that caloric restriction could extend health span by reducing risk factors for age-related diseases. We can think of health span as the warranty period of human life. Just as we prefer products with longer warranties that function well throughout their lifespan, the goal is to extend the period of healthy, active life rather than simply prolonging life at any cost. Ramakrishnan argues that focusing on health span aligns better with both individual desires and societal needs. It could reduce healthcare costs, increase productivity, and improve overall quality of life for aging populations. The book also discusses the role of lifestyle factors in aging and longevity. While much of aging research focuses on genetic and molecular mechanisms, lifestyle choices play a crucial role in determining how well we age. Factors like diet, exercise, sleep, stress management, and social connections all impact our health and longevity. Many of these lifestyle interventions work by influencing the same biological pathways targeted by more high-tech interventions. For instance, regular exercise has been shown to improve mitochondrial function, enhance autophagy, reduce inflammation, and even increase telomere length, all processes implicated in aging. A study published in the journal Circulation found that regular exercise was associated with longer telomeres, potentially adding years to life expectancy. Similarly, a healthy diet rich in plant-based foods can modulate nutrient-sensing pathways in ways similar to some proposed anti-aging drugs. The Mediterranean diet, for example, has been associated with reduced risk of age-related diseases and increased longevity. We can think of these lifestyle factors as a form of everyday longevity hacking. Just as a well-maintained car will last longer, a body supported by healthy habits is more likely to age well. Ramakrishnan emphasizes that while cutting-edge aging research is exciting, we shouldn't overlook the powerful impact of these more accessible interventions. He suggests that combining healthy lifestyle choices with future medical advances could have synergistic effects on longevity and health span. The book explores the fascinating concept of hormesis in aging. Hormesis refers to the beneficial effects of low-level exposure to stressors that would be harmful at higher doses. In the context of aging, hormetic stressors can activate cellular stress response pathways that enhance repair mechanisms and promote longevity. This concept helps explain why some seemingly harmful exposures can have paradoxical beneficial effects on lifespan. For example, moderate exercise causes short-term oxidative stress, but this triggers adaptive responses that ultimately improve cellular health and longevity. Similarly, some of the benefits of caloric restriction may come from the mild stress it places on cells. A study published in the journal Cell Metabolism found that alternating between fasting and feeding activated stress response pathways that improved metabolic health and extended lifespan in mice. We can think of hormesis like weightlifting for your cells. Just as lifting weights causes minor muscle damage that ultimately leads to increased strength, small amounts of cellular stress can toughen up cells and make them more resilient to aging. Understanding hormesis has important implications for developing anti-aging interventions. Instead of simply trying to eliminate all cellular stress, some interventions might aim to induce mild, beneficial stress responses. This approach could potentially harness the body's natural defense mechanisms to combat aging. The book also addresses the role of inflammation in aging, a phenomenon often referred to as inflammaging. Chronic, low-grade inflammation is a hallmark of aging and contributes to many age-related diseases. This inflammation can result from various sources, including the accumulation of senescent cells, mitochondrial dysfunction, and changes in the gut microbiome. It creates a vicious cycle where inflammation causes cellular damage, which in turn triggers more inflammation. Studies have shown that older individuals generally have higher levels of inflammatory markers in their blood, which correlate with increased risk of age-related diseases and mortality. For example, a study published in the Journal of the American College of Cardiology found that higher levels of inflammatory markers were associated with increased risk of cardiovascular disease in older adults. We can think of inflammaging as a slow-burning fire in the body. It may not be immediately noticeable, but over time it can cause significant damage to various tissues and organs. Targeting inflammation is a key strategy in many anti-aging interventions. This can involve directly reducing inflammation through drugs or lifestyle changes, or addressing the root causes of inflammation, such as removing senescent cells or improving mitochondrial function. The book explores the fascinating world of the microbiome and its potential role in aging. The microbiome refers to the trillions of microorganisms that live in and on our bodies, particularly in our gut. Recent research has revealed that the composition of our microbiome changes as we age, and these changes may contribute to various aspects of aging, including inflammation and declining immune function. Studies have shown that transferring gut microbes from young mice to old mice can improve cognitive function and increase lifespan in the older animals. In humans, the microbiomes of centenarians often contain unique bacterial species that may contribute to longevity. A study published in Nature found that the gut microbiome of centenarians contained higher levels of certain bacterial species capable of producing unique secondary bile acids, which may contribute to their longevity. We can think of the microbiome as an additional organ in our body, one that changes and potentially ages along with us. Just as we might transplant a young organ to an older person, manipulating the microbiome could potentially provide rejuvenating effects. Ongoing research is exploring the use of probiotics, prebiotics, or even fecal transplants as potential anti-aging interventions. However, much more research is needed to understand the complex interactions between our microbiome and the aging process. The book delves into the role of cellular senescence in aging. Cellular senescence is a state where cells stop dividing but remain metabolically active, often secreting harmful inflammatory factors. As we age, more and more of our cells enter this senescent state. While senescence initially evolved as a protective mechanism against cancer, by preventing damaged cells from dividing, the accumulation of senescent cells over time contributes significantly to aging and age-related diseases. Studies in mice have shown that selectively removing senescent cells can extend lifespan and improve various markers of health in old age. For instance, a study published in Nature found that eliminating senescent cells in mice could extend their median lifespan by 20% and delay the onset of age-related disorders. This has led to the development of senolytic drugs that specifically target and eliminate senescent cells. Early clinical trials in humans have shown promising results, with senolytic drugs improving physical function in patients with idiopathic pulmonary fibrosis, a age-related lung disease. We can think of senescent cells as troublemakers in a neighborhood. A few might not cause problems, but as they accumulate, they can create a toxic environment that affects even healthy cells around them. While removing senescent cells shows potential as an anti-aging strategy, it's not without risks. Senescent cells also play important roles in wound healing and embryonic development, so completely eradicating them could have unintended consequences. The book also explores the potential of NAD+, nicotinamide adenine dinucleotide, boosters as anti-aging interventions. NAD+ is a crucial molecule involved in many cellular processes, including energy metabolism and DNA repair. Levels of NAD+ decline with age, and this decline is thought to contribute to various aspects of aging. Boosting NAD+ levels has shown promise in animal studies for improving health and potentially extending lifespan. Studies in mice have shown that supplementation with NAD+ precursors, like NMN-nicotinamide mononucleotide or NR-nicotinamide riboside, can improve mitochondrial function, enhance DNA repair, and even reverse some aspects of aging. For example, a study published in Cell found that NMN supplementation in mice could reverse age-related decline in blood vessel health. We can think of NAD+ as a vital fuel that powers many of the cell's maintenance and repair processes. As we age, this fuel becomes scarce, leaving our cellular repair systems underpowered. While animal studies are promising, more research is needed to determine the long-term effects and optimal use of NAD+ boosters in humans. Early human trials have shown some promising results, but larger and longer studies are needed. The book concludes by considering the broader implications of aging research and potential life extension. Ramakrishnan raises important ethical and societal questions about radical life extension. These include concerns about overpopulation, resource allocation, inequality, and the potential psychological impacts of greatly extended lifespans. For example, if life-extending therapies are expensive, they could exacerbate existing health disparities. Additionally, longer lifespans could strain pension and healthcare systems that are already struggling to cope with aging populations. A study published in Nature Climate Change suggested that life extension could significantly increase carbon emissions and exacerbate climate change unless accompanied by substantial lifestyle changes and technological advancements. We can think of radical life extension as a society-wide upgrade. While it offers exciting possibilities, it would require rethinking many of our social, economic, and cultural systems. Ramakrishnan emphasizes that these challenges don't negate the value of aging research. Instead, they highlight the need for thoughtful consideration of how we implement anti-aging interventions and structure our societies to accommodate longer lifespans. In conclusion, Why We Die provides a comprehensive overview of the current state of aging research, from evolutionary theories to cutting-edge experiments. The book makes clear that while we've made significant progress in understanding the biology of aging, developing safe and effective interventions to significantly extend human health span and lifespan remains a formidable challenge. The rapid pace of discovery in this field offers hope that we may eventually be able to delay or prevent many of the detrimental effects of aging. However, Ramakrishnan's balanced approach, acknowledging both the potential and the limitations of various anti-aging strategies, provides readers with a nuanced understanding of this complex field. He emphasizes that while the quest to extend healthy lifespan is noble and potentially transformative, it must be pursued thoughtfully, with careful consideration of its broader implications for individuals and society as a whole. As we continue to unravel the mysteries of aging, we must also grapple with the profound questions it raises about the nature of life death, and what it means to be human.